moved it. Okay. All right. Thank you all for being here. Um, it's my pleasure to introduce tonight's speaker. Adriana is a California native from Oxnard, and she attended UC Santa Barbara. Originally interested in ethnobotany and was interested in studying women's puberty ceremonies, combining religious studies and Chicano studies. And she changed direction after she took Sue Mazur's plant diversity course. And after graduation, she volunteered at the Santa Barbara Botanic Garden and was drawn to their research program, got a job there as a conservation technician when the new beautiful conservation center opened. She decided to continue her work in going to graduate school, and she wanted to stay in California. So she accepted a position with Chelsea Specht at UC Berkeley, and Chelsea promptly moved to Ithaca, New York, which is where Adriana is speaking to us from. And she has been working on Calochortus, as you'll hear. She's interested after she finishes in getting back more into conservation. I also want to say that Adriana is the recipient of numerous awards, including the incredibly prestigious NSF Graduate Research Fellowship Program. She's also deeply involved in mentoring, outreach, recruiting, reaching out to members of the Hispanic community. She's a member of the Diversity Council, the Graduate Student Council, vice president of the Plant Biology Graduate Association. She's acted as a recruiter at the SACNAS and similar conferences and roles for professional societies. She's been involved in public outreach, tutoring, science communication, and it's amazing to me that she actually has time to do all of these things and still get some work done. Now, on that note, let me turn it over to Adriana.
Oh, thanks, Amy, for that great introduction. First of all, I'm really happy to be here. I really like the fact that kind of the silver lining of COVID is that you get to present when you otherwise wouldn't. So yeah, can you all see my screen okay? Great. So today I'd like to talk about the first chapter of my dissertation. I'm, as Amy said, a PhD student with Chelsea Specht at Cornell University. I'm going into my, actually 4.5 years in. And so my first chapter was really looking at one species, Calochortus venestus, which, if you know it, is extremely polymorphic. So I'd kind of like to, going into how to trace its floral traits, evolution of floral traits, and using some population genetic and phylogeographic tools, we uncovered a parallel local adaptation. So first I want to start by going into, really foundational, kind of foundation of evolution, which is natural selection, right? And so in order for natural selection to take place, you need variation in a population, variation of individuals and their traits. And those traits must be inherited genetically, of course. And there must be differential fitness where some of those traits have an advantage over others in the population. And in order for all of that to matter, there needs to be competition or some sort of resource limitation. Right, and so really what I want to highlight here is that very foundation on which evolution happens starts with a variation in traits. So we expect individuals within a population to undergo similar selection regimes, especially when we're talking about within species selection, and that leads to a homogenization of traits typically. Two ways in which that happens is directional selection or positive frequency dependent selection. So for directional selection, here on the x-axis, you see a continuous trait and the frequency of that trait on the y-axis. And we see that this continuous trait has this kind of broad range exemplified here. Under directional selection, there's this kind of preference for this spectrum of the continuous trait, and eventually that frequency of that trait will increase. Under positive frequency dependent selection, we have two traits here in blue and green. And as one trait goes up in frequency, the other trait goes down, right? And so both of these things are leading to this kind of homogenization of trait variation. So I ask, what are the mechanisms that overpower these processes, those that drive and maintain diversification within populations? So diversification is happening at the level of populations. A lot of us, when we learn about divergence or speciation, or you're taught about of allopatric speciation, whereby this kind of original founding population migrates and expands, and there might be gene flow between them, but over time we'll come across a geographic barrier, be it a mountain or a lake or some other kind of unsurmountable body or volume of something. And eventually with time, those populations will split and become reproductively isolated. And so we then consider these two, what were populations, to be species. So work has largely focused on the maintenance of trait variation, either among species or between populations. But fewer studies have been able to focus on within population variation. And one of the ways in which it can be done is typically in hybrid populations. Here's an example of Naudiplicus aurantiacus with a red subspecies and a yellow subspecies. And in this hybrid zone will produce these kind of orange colors. And so studies are often limited to these kind of hybrid zones. And so when you have the opportunity, such as with Calochortus venustus, to study a system in which we believe them to be one species, and they're polymorphic within one population, then you take that chance. You take that opportunity. So I'm going to talk to you about how I'm developing Calochortus venustus as a model system to study floral phenotypic diversity. So first I'll go into how I characterized phenotypic diversity regionally. And then I'll talk about population genetics and how these populations have diversified across space. I'll also go into inferring historical range expansion, again, through phylogeographic tools. And lastly, I'll touch on some of the ecological mechanisms that may be maintaining that diversity. Oh. Skipped one ahead here. Okay, so when I started this project, as many of us do when we don't know very much about a system, we go to the flora, right? So here I have a kind of screenshot of the Jepsonian flora. And what I knew was that petals ranged in color from white to yellow, purple, pink, or dark red. There was this kind of central dark blotch. Generally there was this distal petal spot. As you can see, these are kind of tulip-shaped flowers. And unlike a lot of Liliaceae, there is differentiation between the sepals and the petals, right? These two whorls of petals. Two whorls of organs in the flower. So one previous study looked at phenotypic diversity. They did a morphometric analysis of something like 40 different petal traits and across most of the distribution of Calochortus venustus, but they did not find any infraspecific grouping. So they did not find any evidence for subspecies or varieties. But some of the defining characteristics among all of this diversity that we use to identify Calochortus venustus in the field is that the nectary is square. It's densely covered in yellow trichomes or hairs. There's a dark red petal spot above the nectary. And again, that distal petal spot is sometimes, although not always, present. This is the only species of Calochortus that has that distal petal spot. So if you see that, you know it's got to be venustus. Also, from the phylogeny, which is not well resolved, it might be a little difficult to differentiate other species from venustus. So I just kind of wanted to show some of the species that are in this comb here in the phylogeny. Venustus is highlighted here with this blue arrow. So Superbus has this kind of crescent-shaped nectary. It does have this dark brown spot in a kind of pale or bright yellow spot. Vestae similarly has this spotting, except the nectary are two crescent shapes side by side. And Catalinae has this kind of dark maroon blotch at the base of the petal. That's the one that we have current evidence to believe is sister to Venustus. But morphologically, I'd say that the one that's most similar to Venustus is Argiosus. Argiosus also has some polymorphisms with petals ranging from this kind of lilac or pink color to a much lighter cream, almost white color. And as you can see, this is a mature petal. It looks very similar to Calochortus venustus. It has this kind of red blotch here. But one way to differentiate it, I have a young petal here, is this has a kind of crescent or chevron shaped nectary, whereas again, venustus has a square or rectangular nectary. From previous findings, I also knew that Calochortus venustus bordered the Central Valley of California. You can see that it's kind of forming this circular distribution here. This is from herbaria records from the CCH. And its elevation ranges from 300 to about 2,700 meters. So I started by surveying phenotypic diversity. Again, from the flora, we knew that the petal color varied, that distal petal spot was generally there, and anther color varied as well. So I phenotyped 177 individuals, which I collected in 2019, and you can see in these large pink circles where those locations were. I also made sure to collect the full elevational range, so from lower to higher elevations as well. And those higher elevations are typically located in the transverse and southern Sierra foothills. So what I found, I'm just going to go ahead and let you marvel at this for a second. Essentially, we found, part of what the florist said, what Peggy Fiedler had described, that petal color range from this bright whites to yellows, pale yellows, purples, pinks, reds, and deep shades of all of those colors. So what I found was that there was this kind of continuous spectrum, right, rather than discrete morphotypes. And so we wondered, is all of this diversity really one species? Here, I just want to point out as well that you can see that petal spots were said to be generally present, and you can see in a lot of these individuals that petal spots did not always appear to be present. So we also found that there was diversity in the degree of polymorphism in populations. So some populations were quite polymorphic. Just within a foot or two apart, you can find individuals of every color. And other populations are exclusively white, right? So very monomorphic. So I started to think about how this diversity varied across regions. And so here I'll have a few slides where I show this larger histogram showing metapopulation data. And on the right-hand side, you could read this as a map. Thankfully, California is shaped in a way that agrees with charts. So this is the coastal populations, these are Sierra populations, and here are the transverse ranges. And essentially, we found that white was the most common phenotype, and magenta, pinks, and reds were very similar in frequency. But when we look at this regionally, we see that those polymorphic populations, again, are almost exclusive to the transverse ranges, a lot of phenotypic diversity in the south. But the northern populations have a lot of white individuals. Sierra North populations, again, a lot of white individuals, and central Sierras as well. So essentially, we saw this pattern of strong phenotypic diversity in the south and much less diversity in the north, almost exclusively white. So I started to wonder about the frequency of these different colors across elevations. So for the yellow here, you can see that it's found pretty much at the lowest and the highest elevations, but more commonly in higher elevations. For the white phenotype, it's almost normally distributed. So we see, a lot of this color in every population. For pinks and purples, we start to see those more deeply pigmented flowers at higher elevations. And red phenotypes we see exclusively in the highest elevations. So we also found that abaxial color varied. This is not something I had read in the flora or really read anywhere online or heard from anyone else. But here I have some examples of, on the left-hand side, this is a white flower. So on the inside it's white, but the outside could be white or pink. Here I have a red, these two are red flowers where the inside is deeply pigmented red and the outside is, here is yellow and here is white. And we saw just such a broad range of diversity of, abaxial compared to adaxial or internal versus external pigmentation. And so at the metapopulation, we found that mostly the exterior did have a different color than the interior. Regionally, that was true for the coastal and southern populations. And in the Central Sierra populations, we actually found the opposite, that typically the exterior interior flower had the same pigmentation. We're not quite sure what these traits mean or if there's any kind of ecological significance yet, but this is really just kind of me characterizing regional diversity. If you remember, petal spots were described to be kind of generally present. And I'd say at the metapopulation scale, we actually found that there was almost a 50-50 of a split between that petal spot being there or not. And we found a regional pattern where the coastal populations did have that petal spot, but the Sierra populations did not. That was statistically significant. So there's this kind of east to west or Sierra to coastal divide where petal spots were almost exclusively found on the coastal populations. We also found that anther colored varied. This is something that Peggy Fiedler described in her flora. We found that almost, most individuals had this kind of yellow to white anther, but there were a high number of pink or purple anthers in the transverse ranges. So we start to kind of see that there was a lot of phenotypic diversity in, again, the southern populations. And lastly, filament color was found to vary. So anything from this, white or colorless filament to a very deeply pigmented filament. At the metapopulation and for most regional, populations, there was no statistical significance between whether or not there was pigmentation in the filaments, except for the Sierra southern populations where filaments were significantly white or had just a tinge of color. So it's so much variability across individuals as well as the traits. I wanted to do a principal components analysis or essentially see how often these traits were occurring together. And I just want to point out here that adaxial petal pigmentation really drove that diversity as well as the presence or absence of that petal spot. And I wanted to look for groupings, again, just to see how these traits were occurring together or if traits were commonly occurring together. And we found these kind of three general groupings. So here, if you had an adaxially white petal, you were likely to have a petal spot, likely to have a yellow-white anther, and likely not to have any pigmentation in the filament. So this is a fairly white flower and is very commonly what we see when we see a representative of Calochortus honestus. We also found this fairly pink flower where the adaxial side was, or the internal petal was pink and the external petal color was some other color, and the filament was also pink. And then this kind of third grouping, a very deeply pigmented flower where either the petals were magenta or red, the anther was commonly purple, pink, and there was no petal spot. And so these kind of, highlight these suites of traits that are commonly occurring together, suggesting that there could be an ecological significance to these traits. So just to summarize this section, adaxial pigmentation is found to be continuous through a spectrum of these colors rather than discrete colors. The abaxial or the external petal pigmentation was also found to vary, and it was often different from the internal petal color. There was phenotypic variability both within and between populations. Again, there were some populations that were very polymorphic while others were almost exclusively white. And that phenotypic diversity varied geographically. So we found this kind of east to west directionality where petal spots were found almost always on the coastal populations and never on the Sierra populations. Okay. Again, I kind of just summarized that quickly. We have all of this phenotypic diversity. I binned them into one of these 5 colors, and it's just a little bit easier to see here that all of this pigmentation or this diversity in pigmentation can be found in the transverse ranges, and we start to lose that pigmentation as we go north. So that to me was suggestive of local adaptation. So I wondered, how have the population genetic dynamics and the evolutionary history shaped the diversification of these traits across the landscape? So I wanted to ask if, again, were all of these phenotypes actually one species, or what, as in was there gene flow between these different forms? And then what is the gene flow among and between these populations? And California is a very large place. So are these mountains acting as corridors, or are there barriers to dispersal? And how did historical range expansion happen? So if populations moved from south to north, we would see this kind of parallel evolution toward the white phenotype, since it's the most common phenotype there. Or if it moved from a north to south directionality, then there may be convergent evolution of the phenotype, where populations have kind of independently derived this pink phenotype or purple phenotype somewhere along the way. So I'm not going to get too heavy into the methods, but I do want to share some details. So we were able to sequence 190 individuals, including some outgroups, and each one of these have phenotypic data. I also want to highlight these 2 students who helped me extract all of that DNA. So Martha Williams and Dariana Argueta Zamora, who are, Martha was an undergrad at Cornell and has since graduated, and Dariana is an undergraduate. So we used RAG sequencing, which essentially cleaves just throughout millions of sites across the genome. And when I started this project, I didn't have a reference assembly. This is a non-model organism. Not a lot of work has been done on it, so much less genetics. It has a very large genome, but with the help of a postdoc, Jacob Landis, who who assembled this genome, we were able to map our sequenced reads. So once we mapped it to the genome, kind of filtered out a lot of noise, anything that sequenced poorly, et cetera, we were left with 94,000 SNPs and 174 samples of Venustus. Okay, so then we assigned individuals to populations, inferred population structure and historical range expansion. So the first thing I want to point out was that there's no clustering by adaxial petal pigmentation. So here's a principal components analysis where each dot represents an individual colored by its petal pigmentation. And if there is, if we expect each color to have its own genotype, or essentially to be its own species, then we would expect clusterings by color. But we see that there is no clustering either at this metapopulation scale or even within subpopulations. Instead, what we see is very strong geographic cohesion. So each one of these kind of general clusters pertain to geographic locations. And so we can see this bifurcation of the Central Valley, where there are 2 populations in the Coast Ranges here and 3 populations in the Sierra Nevada foothills, with this highly diverse population in the transverse ranges. We also inferred ancestral populations, or essentially assigning individuals to a population and kind of assess admixture. And so you can test different numbers of populations, or what we call K equals 2 through 7 is pictured here. Really what I want to point out is just how these populations are structuring as we increase the number of populations. So when we have a model of K equals 2 or 2 populations, we see a bifurcation between the Coast Range populations and everything else. And at K equals 3, we start to see a divide between transverse ranges and the Sierra Nevadas. At K equals 4 and 5, we start to see divergences along the Sierra Nevadas. And at K equals 6 and 7, we start to see more divergences across the Coast Ranges. So how do we know what number of populations best describes our data? I used a cross entropy criterion, essentially just looking for the lowest number on this plot, and that was K equals 5. So when I look at my structure or admixture plot at K equals 5, we see that there is that concordance of population structure based on geography, and it looks very similar to our principal components analysis. But we do see admixture between neighboring populations. So for example, here are the Coast Ranges, and we see that there are some alleles shared between the transverse ranges in purple and the Southern Sierra Nevada foothills in blue. This is a very highly admixed group pertaining to this kind of these populations here in Carrizo Plain going into Paso Robles. And again, we see that there's a lot of shared alleles with the transverse ranges, the Southern Sierra Nevada foothills, and in light gray, the Coast Ranges as well. And you can kind of see this, almost perfect correlation between geography and genetics. So in pink are the Northern Sierra foothills. Here and here are the transverse ranges. And you can see that with increasing geography, you see increased genetic similarity. So then we did an explicit test of essentially FST or allelic heterozygosity between populations compared to within population heterozygosity. Essentially found that same trend that there was increasing genetic differentiation with increasing geographic distance. And the kind of most important thing we saw here is that there was the largest genetic differentiation between northern populations, which we kind of expected based on the distribution, with very few individuals in the Central Valley, most of which are historic collections. So we explicitly tested for isolation by distance. Again, this trend of geographic distance compared to genetic distance. And at the metapopulation scale found that strength was very weak. That trend was very weak. But we know that genetics and geography are heterogeneous across the landscape. So I tested for isolation by distance within these major mountain ranges. And for example, in the transverse ranges found, again, a somewhat weak trend of isolation by distance, although it is statistically significant. In the Sierra Nevada ranges, it was quite a bit stronger at 0.22, and the Coast Ranges was the strongest at 0.55. So generally, I think we gained a lot by looking at these trends at more regional scales. So another thing we were able to do in order to kind of assess migration or gene flow is this method called effective migration surfaces, where essentially you assign individuals to a population, and based on their, you compare their genetic distance to their geographic distance as a proxy for effective migration. And these, it's really just a visualization tool where you could think that under a null model of isolation by distance, you would find these regions pictured in white. And if you have much higher migration than average, you would have these regions pictured in blue, and much lower migration than average would be pictured in orange. Essentially, this is helping with the identification of any kind of barriers or corridors to migration. What we found for Calochortus funestus, well, again, first I want to show you here there is migration on the left and diversity on the right. And we were able to reconstruct the Central Valley, right, where we don't expect any or much gene flow, historic gene flow. We also found that the west of the Sierra foothills could act as a corridor here, or may have historically, but currently the Central Valley is likely very unsuitable habitat. Then in the kind of southern end of the ring here, we see that the transverse ranges to the southern Sierra Nevada foothills are acting or have acted as a corridor historically. So you can also assess diversity in this way, and found that there was high genetic diversity in the transverse ranges and low genetic diversity in the coast ranges. So to kind of summarize this section, first, we do see gene flow between color forms, right? So it doesn't look like each color has its own genotype, which affirms this concept that Calochortus venustus is one species. And the metapopulation is generally structured into 6 subpopulations, which in general have differentiated through isolation by distance, yet they remain interconnected through these 3 major mountain ranges, which we saw are acting as corridors. And there's admixture between our neighboring populations. The strongest genetic differentiation is in the northern populations, and the southern end of the ring holds the highest genetic diversity, as well as phenotypic diversity, right? So now going into historical range expansion and directionality. Again, not going to go into details about the methodology, but we inferred a maximum likelihood tree in MaxML and applied an ascertainment correction, typically because you get really long branches when you use SNPs. So you can provide the counts of invariant sites for each base pair. So first I want to say that Calochortus venustus is monophyletic, and so are our outgroups. So we have Argiosus, about 4 or 5 individuals of Argiosus came out monophyletic, and sister to Catalinae. And that our tree is generally consistent with our population genetics analyses, right? So our these clades are not formed by petal morphotype. I have here each clade with its kind of individuals represented, the phenotypes represented by these color blocks. And so if each phenotype had its own genotype, or if they were different species, for example, then we would see kind of a clade of pink, a clade of purple, and so on. And we don't see that here. So instead, I'm going to walk you through how these clades bifurcate, so each bifurcation and where it maps to on the landscape. So the first bifurcation are these individuals in the Carrizo Plain, suggesting this is kind of the general area where Venustus diverged from its sister species. The second divergence is in the coast ranges. And we see that, again, mapped here. And one thing I want to note, just that there are really short branch lengths here, suggesting there was a possible bottleneck from our Eames analysis, those historical migration and corridor assessments. We saw that there was low genetic diversity in this population as well. And there was this kind of general barrier to gene flow there. So that's kind of, concordant analyses with each other. Another bifurcation is in this kind of central coast range population. So we see that there was this kind of back and forth migration in this area. Then we start to see a range expansion happen toward the east, here with the transverse ranges. And with the southern Sierra Nevada foothills, we see kind of 2 clades diverging. So again, there was this kind of back and forth migration in this area here. And finally, we see populations kind of moved north, right, in the green and in the pink here. And so together we can see that there was this bidirectional, as in this kind of 2-way coastal and eastern migration from south to north. And there was strong geographic cohesion. So together with our phenotypic data, we can see that there is this parallel evolution toward the white phenotype, right? So these populations are almost exclusively monomorphically white, suggesting that there is a selection pressure for that phenotype and that there's a local evidence of local adaptation. So just to kind of summarize that, we found that Calochortus venustus is monophyletic. These clades were not formed by phenotype, but there is, gene flow between each color form. There is a south to north bidirectional range expansion, giving us evidence of this parallel evolution. And the north coast holds low genetic diversity together with the fact that there are short branches, suggests that there is a possible historical bottleneck. So just because I'm running a little short on time, you know, with all of this phenotypic diversity, these kind of spatial patterns, regional patterns, the fact that there was this east to west differentiation of that petal spot, that there is gene flow between these various color forms, you know, all of this is suggesting that these petal traits are suggestive of local adaptation. They're evidence of local adaptation. And so I started off asking, how is phenotypic diversity maintained in a population? And, you know, unfortunately, or actually maybe rather fortunately, I don't have an answer to that yet. So I'm still looking. And so it's likely that it's a heterogeneous landscape of environmental factors, right? So biotic and abiotic factors that are contributing to that distribution and then evolution of different forms. And so I've started to answer that by using species distribution models to identify kind of the important physical environmental variables that are associated with each phenotype. So I'm running a model for each color, you know, red, yellow, purple, pink, and white. And then we can also use that to kind of assess whether fitness or dispersal is limiting the distribution. And so I'm going to show you on the next slide one of these for each color form, one of these graphs. The one on the left is variable contribution. So on the x-axis is the percentage or the importance of each of the variables that are on the y. And so for this one, we see that this variable, which is bioclimatic variable 19, is greatly contributing to the model that causes this distribution of whatever species this is. And then this map, which is a probability of finding this species across the landscape. So with a high probability in green and a low probability in white, or this kind of tan color. So there's a lot of data on here, and really without getting into too much detail, I just want to go over some of the broad strokes. So some of these morphotypes, the pink, red, and yellow, are primarily correlated with temperature variables, while the white and purple phenotypes are more strongly correlated with precipitation. We also find that some of these phenotypes have a significantly smaller distribution than we would expect under the model. That is the red and the yellow, where we see that there are these kind of other patches where we don't find these phenotypes, suggesting that they may be limited by dispersal rather than fitness. So some of the future directions, again, I'm looking to continue answering what is maintaining within population diversity. So I'm doing some ongoing work with pollinator observations, trying to correlate each phenotype with a pollinator presence, abundance, as well as constancy or their behavior. I'd like to survey the abundance of morphotypes across the season to investigate any possible phenological displacement, and evaluate any differences in seed set between morphotypes, which could be an indication of frequency-dependent selection or temporally varying fitness. And with that, I'd like to thank the Spec Lab. We're pictured here at Botany 2019, back when we could travel. My committee, who is Monica Gaber, Kevin Nixon, Rob Raguso, and Chelsea Specht at Cornell. Various people at Cornell who have helped me with this project. Peggy Fiedler, Danette Sines, and my undergraduate mentees, and of course, my funding sources. Thank you.
There. Wow, that was an incredible amount of information. That was really phenomenal. Let me show my face again here. Hold on a second. Thank you so much. I can't believe that is a really, really complex and fascinating system. I will be happy to throw the floor open for questions if people want to either put questions in the chat or see if you can unmute yourself. I don't see any questions in the chat so far, except Adriana, are you now at Cornell? Does anyone want to ask questions? Do you think it could be this is from Justin Long. Do you think it could be herbivory or disease limiting the range instead of dispersal? Or is there a particular reason you think it is dispersal?
Yeah, so I mean, dispersal for that yellow and those white morphs, sorry, yellow and red, that's really just what the climate variables are telling us, which is a very limited amount of information. So I don't really want to answer that yet without looking at seed set, pollinators, and some other things. I will say though that what we found with the red pigments being exclusively found at higher elevations, and knowing that that red pigmentation, which is caused by anthocyanins and is also known to protect against ultraviolet radiation, might be at play. So we might see that increased pigmentation because they're protecting against, again, UV rays.
A couple more questions here. Nancy Morin wants to know, have you looked at UV patterns in the flowers?
Yeah, I tried my first year. I didn't see anything, different when I put that UV light on there. But I didn't really do it systematically. I looked at like one population, maybe 4 or 5 flowers. So I have plans to do that this upcoming field season.
Cool. That could be really interesting also. Amanda Fisher wants to know, what are the different pollinators would you expect on visit C. venustus?
Yeah, so very little work has been done in this system, but one of them was on pollinators, and they actually did a pollinator study on, I think, all or a lot of Calochortus species. And they found kind of generalist pollinators, so bees, beetles, just kind of all across the board. But they did not differentiate between color forms. So we know what pollinates the species as a whole, but we don't know what pollinators, visit each morph. I feel like the pollinators is what I know least about, but you might expect with that red pigmentation, you might see some flies, but I haven't really noticed any trends in my field work. So yeah, we'll know this upcoming season, hopefully.
I just allowed you to unmute yourselves, I think. So if anybody wants to actually ask a question out loud, you can do that. I'll wait a second here and see if anybody wants to. No? Okay. Well, Fenerty family says, do any other Calochortus species show such a wide range of color?
No. No, I mean, there are a lot of polymorphic species. And you might see fairy lanterns that are, a lot of, like Calochortus albus is white, but there are some red morphs. Weedii and Plumerae and, that kind of whole clade has a lot of variation. Argiosus, like I said, there's pinks and purples and whites, but I think this kind of full spectrum is really something we only see in Calochortus venustus.
It's really pretty insane, actually. I have a couple of questions. One is, have you thought about looking to see which genes your SNPs are in, to see if you see any association with environmental variables, for instance, or geography, or?
Yeah, that's a great question. That's moving forward. So our current reference genome was really just to map these reads, these SNP reads. So we have to build on that reference genome, and we need a lot more coverage to get to that point of being able to associate loci to actual functions. So we actually, speaking of that, have a grant in to do that. So I hope in the next year that we'll have a really good, well-polished assembly and can do some of those, detect some of those loci under selection.
Awesome. Also, have you thought at all about, I'm sure you have, thought a little bit about the role of epigenetics in all of this?
Yeah, yes. Yes, and if I guess I just want to say this, I'm not going to do it in my PhD. I really am kind of trying to develop the system as a model to study the evolution of floral traits and pigmentation. So that's something I'd consider doing forward. I think it very well could be epigenetic. Yeah.
Unfortunately, there's so many different mechanisms of epigenetic regulation, some of which are easy to study and some of which are difficult. So it's not necessarily that straightforward anyway. But it would be nice if it were easier to study, because I think it probably plays a role in a lot of these phenomena like this. I'll stop sorry, my cat is. Anybody else have questions?
Thanks everyone for the kind comments. And if you ever have a question or a comment, I'd be happy to, get your email and chat.
If you want to just unmute yourself and say hi, I think you can do that at this point. I guess everybody's just super shy here.
I'd love to see your faces. I recognize a lot of names. Oh, a question about misidentifications. Yeah. I plot the herbaria records. Oh, thank you. Yeah, I show the herbaria records on the map because there are so many iNaturalist observations that are incorrect IDs. And that's actually part of why I include that, the first few slides showing species that are in that clade, and you can fairly easily differentiate them once you take a look at the nectaries. So yeah, there are a lot of mis-IDs. Mm-hmm.
Anyone else? Should we let her go to sleep since it's almost 11? That was really, really great. I got so many ideas from it, and it's just such it's just such an incredibly complicated system, and it's just so bizarre that you have just this one species with so much variability. My cat thinks it's dinner time. Sorry.
Yeah, I think it's made me think about things in a different way. Typically when you're studying one species, you're thinking about like metapopulation scale. And so having so much diversity kind of forced me to look at things more regionally, more at the subpopulation level. And I think that's what a lot of studies could gain from as well.
I have some other thoughts too, but I'm going to send you an email. So yeah,
great.
So my cat wants dinner. That's what he wants. So anyway, okay. I'm going to mute myself and thank everybody for coming, and we'll see you all in a month. Thanks very much. We'll put the video up on the website. Take care. Great job. Rachel, did you want to say something?
I've never met you, Rachel. It's nice to see you.
Yes, nice to see you. So sorry, I was kind of in and out, just getting my little guy to sleep, and it's, as you can see, a crazy mess here. But a wonderful talk. Thank you so much for doing this.
Thank you for inviting me, really. I appreciate it.
Yeah, I'm so glad it worked out.
Yeah.